Hello students. So today we will try to learn about these cells of the immune system. So basically we can divide the cells of immune system in two types of cells. The cells which are contributing towards the innate immunity and the cells which are contributing in the adaptive immunity. So first we start with the cells which are contributing in the innate immunity. But one should be clear that both innate immunity and adaptive immunity, they are communicating with each other and they are coordinating with each other and they are getting the same immunity, or the goal they will achieve is immunity. So we cannot totally say that macrophages are contributing towards innate immunity, but there are antibodies which will inform the macrophages, and we know antibodies are the part of adaptive immunity. So first are the macrophages, and macrophages are doing this phagocytosis and antigen presentation and cytokine secretion. So the function of these macrophages is that they will kill pathogens, microbes, and through this phagocytosis and antigen presentation, that process antigen will bind with MHC II and will secrete cytokines, and those cytokines will also target the pathogens or pathogenic cells or bacteria or viruses. So tissue, where they are, so tissue resident forms, for example in the brain they are termed as microglia, and in the liver they are termed as Kupffer cells. So here you can see this figure in which you can see that these macrophages have these dendritic cytoplasmic extensions, and which are termed as pseudopodia, and they have phagosomes and lysosomes. Lysosomes will fuse with phagosome and will make phagolysosome, so that whatever is phagocytosed in the form of phagosome, it will be digested and with help of hydrolytic enzymes of lysosome, and then it will be processed, and these processed proteins will be presented to the Th cells with the help of antigen presentation process. So here you can see in this antibody dependent cell mediated cytotoxicity response. So macrophages are secreting these two types of molecules, that is lytic enzymes and tumor necrosis factor, which will target the cell and will kill the target cell. Next we come to the second group of cells, that is dendritic cells. And dendritic cells are the cells which are mostly present in the thymus, and they are contributing towards this T cell selection, T cell maturation process to educate the T cells, so that they will be so that our immune system will be able to differentiate between self and non-self, so that it will not react from against the self proteins and will act against these foreign particles or pathogens. So dendritic cells, most potent antigen presenting cells, and bridge innate and adaptive immunity, and migrate to lymph nodes to activate T cells. So here you can see this dendritic cell is more or less similar to the macrophage, but it has more cytoplasmic extensions, and you can see that it's written that it will migrate to the lymph nodes, and also it will be in the most of them will be in the thymus. Neutrophils. So neutrophils first responders to infection, and they will do this phagocytosis and will do degranulation. Degranulation means that they will release some substances which will be active against pathogens, and netosis, neutrophil extracellular traps. So neutrophils, here in this diagram you can see that these neutrophils are secreting these lytic enzymes to destroy target cell, and these neutrophils will be informed. Neutrophils and these NK cells and eosinophils and macrophages, all these will be informed by the antibodies, because these cells have receptors for the Fc part of the antibodies. So antibodies will inform these all four different types of cells about the target or the danger or the pathogen, so that they can act and they can kill that target cell or pathogen. So the first responders to the infection are the neutrophils. It means when there will be infection, then the population of neutrophils will increase. That we can determine with the help of DLC, differential leukocyte count technique. Eosinophils. So eosinophils that combat parasitic infections and role in allergic infections. So eosinophils combat parasitic infections, and it's play a role in allergic reactions. Basophils release histamine and other mediators during allergic responses. So it have these granules in the packets, and they will be released when there will be this allergen will come, and then allergy response will be produced. But the most active one in the type 1 hypersensitivity are the mast cells. So mast cells reside in tissues, mediate allergic inflammation and anaphylaxis. So it will mediate allergic inflammation and anaphylaxis, and they have these packets which are filled with histamine, and when this IgE when IgE will stimulate, here you can see that these mast cells, on its surface there are these receptors for the IgE. So IgE will bind with the help of Fc parts with receptors, and allergen will come and it will cross-link these two antibodies which are bound with this mast cell, and then it will release histamine, and that histamine will act on target cells and will produce allergy symptoms. So here is a detailed overview of mast cell presented in bullet points. So these mast cells are derived from hematopoietic stem cells in the bone marrow, enter the bloodstream as immature cells, migrate to peripheral tissues, skin and mucosa, and mature in situ under the influence of local factors, stem cell factors, and belong to the myeloid lineage of immune cells. So there are two lineages of cells which will develop from the hematopoietic stem cells: lymphoid lineage and myeloid lineage. So from lymphoid lineage, it will make these T cell, T C cells, and B cells, and from myeloid lineage, it will make these maybe red blood cells, megakaryocytes, and also it will make mast cell. Location in the body: reside in connective tissues and mucosal surfaces, that is skin, respiratory tract, and gastrointestinal tract around blood vessels and nerves, act as sentinels at sites exposed to the environment. Structure: large round cells with a central nucleus. Cytoplasm contains numerous basophilic granules. Granules stand strongly with toluidine, blue, and other basic dyes. So these granules contain mostly histamine, increases vascular permeability, causes smooth muscle contraction, and will produce type 1 hypersensitivity response. Heparin, anticoagulant, proteases, tryptase, and chymase, and cytokines